Hi. So far we have been discussing a lot about the cellular part of the story and neurons and how neurons function and things like that. We have been looking at the functions of the nervous system in general. I also gave a brief idea of how the nervous system is organized with reference to the human nervous system. I told you about top level stuff of very gross distinct areas such as the brain, spinal cord, which are very located at various different locations. So, the next step is to understand a lot more about the anatomy. Now, why am I focusing so much about anatomy? When I designed this course and thought about the syllabus, my initial idea was not to incorporate too much of medical stuff into the program. But there were a couple of issues which I later thought about, and I think you should be aware of that. As it stands now, neurosciences is necessary and used by a lot of very diverse group of people in their work. And as in my introductory class, I had told that the course is designed for all of those. Now, the advantage of an online course is you are not restricted to a particular syllabus, and which the freedom of which I have taken so far. So, it does not follow a medical curriculum, so in the sense that very rigid compartmentalization of topics has not been followed. I have taken a natural flow which basically assumes that you have something like 12th class 12 knowledge, or maybe much younger, because this is class 12 of when I was doing 12th. So, people study a lot of this stuff in much earlier classes these days, I have seen. So, the idea was to build it from there on and build it without giving a medical flavor as much as possible, even to the extent of when I have been choosing headings for topics, I have tried to make it as non-medical as possible. But having said that, it is not possible to give a complete non-medical flavor, because a lot of stuff is biological, medical, and it is what it is. You cannot offer an apology to that and make it more simplified than it needs to be. So, the idea here is to make it palatable, that I think is the goal of the study. So, I have titled this as Build Brain, which you would understand in the next couple of sessions on why the title is so. And it's a new effort from my side, because I had to explain the brain to a very novice audience, which I presume it to be. And I also have to ensure that you have competence. Now, I use the word competence for a very different reason. So, I have come across students who use the brain anatomy and brain function in their studies, engineering students. So, the most distant group is the IT group, who use very nicely the terms CNNs, RNNs, LSTMs, and the people who design these packages have been using core neurosciences concepts. So, even starting from a CNN, so CNNs have been inspired by the visual pathway and the visual cortex. So, if you are working with CNNs and you are very appreciative of the work which a CNN does, it would be nice if you could know the nervous system in greater detail. On the other hand, there are students and faculty who are working with brain images for various kinds. It can be for image processing, it can be for finding out, for annotating, for various kinds of studies where brain images are used. Now, what happens in such kind of work is that the introductory class is basically about under an hour. So, in under an hour, neurosciences brain anatomy is difficult to be taught. It is possible, but you obviously compress data beyond a particular resolution, and at which point it becomes useless. So, I do find students struggling because they do not have an intrinsic understanding of brain anatomy, but you are forced to work with brain images. So, it can be normal, it can be tumors, it can be several other kinds of diseases, but it is finally brain anatomy. So, they are instances of anatomy or disease states which you have to process. The same thing holds for even signal processing with any kind of electrical data, that is EEG, EMG and things like that, where you do not start with the signal. You should have an understanding of the background from where it is coming from, which I have tried to not the where part of it, the how it is coming from, which I have dealt to some extent. But better understanding of where it is coming from is also useful, because it put a lot of context into the stuff which you are working. So, it is with that idea. Now, how do I bound it? How do I bound an anatomy class? Do I make you competent enough to go to a neurosurgical operating room and perform surgery? Well, that is not exactly an idea, but for somebody who literally works on a daily basis in the inside of the skull. So, it gives me a unique opportunity to share a different perspective and viewpoint. Now, as I told you, even if you look at medical curriculum, neuroanatomy is a very difficult topic. You ask any first year MBBS graduate, first year MBBS student, you would find that neuroanatomy is not something which is very popular. You can have an intrinsic interest for that, but if you look at the section on neuroanatomy in Gray's Anatomy, which is the standard anatomical textbook, it is very dense. It becomes worse when you look at books like Carpenter's neuroanatomy, which is so complex that you would feel intimidated on even holding the book and reading it. So, these are two extremes. So, you have material which is very difficult to grasp, very difficult to acquire, and you have material which is too less in the sense that it does not become working competence. So, I had to find a bridge between these two extremes. The bridge most often may not be simple. So, that is why this preamble to the discussion, and that is what I am trying to do. So, teaching anything is one of my collaborators would call it as a performance, and as in all performances, there is some message to be conveyed, whether the media can be different. Here we have it online and stuff, but even in an when you have a face to face audience, it is important to convey a certain meaning to that. And the beauty of teaching is that you are free to experiment with stuff. So, there is a lot of experimental stuff which you are going to see, which I am experimenting, because I feel that I tell you that the optic tract is superolateral to the midbrain and goes and joins into the colliculi, and from there goes to the primary visual cortex on the in the occipital lobe, and from there goes on to Wernicke's area. That by the time I finish my first half of the sentence, you have lost track, and it is expected to also. So, what you are going to see is a very different kind of experiment. It is an experiment from my side in trying to teach a very diverse audience such as is expected for the course. So, please do bear with me. I am not sure whether it is going to be a success or failure, because unlike in a live audience, you can't gauge from the audience reactions whether it is interesting, not interesting, whether it is possible to grasp, or it's difficult. So, a flavor which I have maintained throughout the course so far is that I've, in spite of my bad handwriting and this one, I have tried very minimal photographs and things like that. So, I am not this is not a class in which you prepare for an exam. It is a class, this is a course in which I try to make you understand things. The objective is not that you finish the course, get the certificate and show the certificate. The complete objective of the course and the time which is spent over here is that the objective is that you understand the stuff. You may not remember the stuff, but you need to understand the stuff. There is some difference. Of course, if you understand well, you would obviously remember, but the vice versa may not be, the contrary may not be true. So, what I mean is you may remember something, but you may not understand it. So that being the case. So, a little more introduction here I think I need to say, because as I was telling you, I had to bound my class. So, we are looking at brain anatomy, and I already familiarized terminologies, that is what is the cerebrum, what is the cerebellum, what is the brain stem. One entity which I am not, these are core neuronal structures, in the sense that these contain nervous tissue, both the functioning neurons and the astrocytes and oligodendrocytes and several other supporting cells which are there within a nervous system. Now, I did not mention many other things, which is, the brain is not just about that, and as neurosurgeons we need to know a lot more about lot more other stuff. But it also follows through that as an audience who is studying brain anatomy, you should not, you can, but you should not take the brain anatomy in isolation, because it is very difficult. Our memories to a large extent are relational in the sense that you relate something to something else, and then you remember both ideally. So, when you are taught A for apple, you remember A in the context of the apple, and you remember that the entity in your hand is named apple. So, the labeling of the topic and the topic itself form internal associations by which we remember both. So that's the principle. So, in the context of the brain, why I have to say that when you start with brain anatomy, I think I have to start with the skull. Then there is something called as meninges. Then you have blood vessels. Now, as I told you, it is not my intention to make you brain surgeons. So, I would limit my discussion on many of these topics, but without having this background, it is a bit foolish to go deeper. We will start with blood vessels. So, blood vessels, as you might be aware so far from your prior education or through my discussion on gyri and sulci, there are something called as artery and vein. Now, apart from vein, we have got something called sinuses. Sinuses are there everywhere. So, there are sinuses around the nose, there are sinuses in the blood, in the brain, and then there is a sinus in the heart, and so that's how it is. So, we follow terminologies with the understanding that an important other entity which you would need to know is ventricles and cerebrospinal fluid. Okay, so these are the supporting actors in the story, and I would like to start out with the supporting actresses. For the very simple reason that when you see you cannot see the brain in isolation, you should not see the brain in isolation, and that is the point. So, these are brief, I would show you a lot more details later on. So, blood vessels, it is also important to understand that these have disease bearing. So, when somebody is studying on stroke, you should have an idea of brain blood vessels. Whether you are taking fMRI data of post stroke patients, or you are looking at annotating stroke areas, early recognizing through AI packages, all those stuff happens, and there are a lot of people working on each one of these things. So, when you are doing an optimization, you should have a basic understanding of what blood vessel and what is a stroke about. No? So, that is the idea of speaking about blood vessels. Now, artery, which is arteries in plural, start from the heart. Okay, and then that goes to the neck, and that goes to the brain. Okay, so there are a couple of vessels which I will deal with it in greater detail. So, this is the general, it is a tree, basically it is a tree. Okay, and it splits. So, the how these blood vessels go through, as I think I told earlier in one of the classes, it is like this, you have the veins on your hand, these blue colored things over on your back of your hand, those are the veins. So, incidentally that is veins. They go in reverse. So, we name them in reverse. So, they go from the brain, and there are things called sinuses, and through that to jugular veins and back to the heart. So, that is the general arrangement. So, this is so arteries go into capillaries and then go into the venous side of the story. So, there are a lot of diseases, there are a lot of stuff which incorporate this simple notation. Now, if somebody is working with angiographic images, angiographic images would know the importance of this. So, there you have phases of these things and then you can find out. It is also important to know the circulation in say circulation time for CT angiography data. So, where you calculate the injection, you synchronize injection of dyes to circulatory time, and circulatory time is the time taken for blood to circulate from one heart cycle to the other heart cycle, and that is important. No, not basically from heart to heart back to heart, but from the heart to the brain and the various phases of the intracranial circulation. So, these are necessary for various kinds of data acquisition protocols, where if you are doing contrast injection, you need to time the contrast to the time of acquisition, and then you do various kinds of studies based on that. So, CT angiography, I am also incorporating some imaging terminologies. In my previous classes, I have already introduced the ideas underlying ideas which are there for functional MRI. I have also introduced the concept of positron emission tomography, the user part of the story, not the physics or the background. And I am introducing the idea of angiography, but which is both digital subtraction angiography and CT angiography here. So, this is something about blood vessels. Now, interestingly, as opposed to many other parts of the body, arteries and veins go separate paths. So, they only meet in the heart. So, so what that means is in most other parts, arteries and veins go together. So, the arterial distribution, the arterial pathways are the same and are same to the venous pathway. So, if you look at the leg, you have got the saphenous vein, the femoral artery, all of them together. then in the arm the brachial artery, the cubital veins, so many of them are quite together and they have similar pathways. In the brain they are pretty well differentiated to the extent that even the gyrus, which is the entity, the anatomical atomic entity within the brain, it has its own different arterial path and a separate venous path, which I have discussed in the discussion on gyri and sulci. So, that is the level of so what happens is arterial territories are different from venous territories. So, when there is a venous damage, the amount of tissue which gets lost is very different from the tissue lost which when an arterial territory is affected. So, those are again things which you would territory is not equivalent to venous territory. Okay, so that is a brief introduction and something basic about the blood supply of the brain. So, the brain is very richly supplied with blood. I have spoken about the Krebs cycle, glycolysis, the power system of the brain, and I have also highlighted the problem with the brain that it does not store glucose. There are no features to store glucose and it is live. So, when there is a higher requirement of glucose, you increase blood flow and it is a very tightly regulated system. So, it also means that the amount of density of blood vessels per unit tissue is very high in the brain and for ensuring that smooth functioning. So, it is very richly perfused and brain tumors as such, operating on brain tumors for that reason is pretty bloody. We encounter blood losses of easily about 1 liter, 2 liters up to 5, 6 liters and things like that. So, for the amount of tissue which is affected, the amount of blood which is lost is disproportionately high. So, that is what I mean. So, that is about the arteries and veins. Now, so that is yeah. Now, we come to something called as meninges. Now, for the sake of this one, you need to know terminologies. So, dura, arachnoid and pia. So, these are the various kinds of meninges, basically coverings of the brain. So, you have dura, which is the outermost cover which covers both the brain and the spinal cord. There is arachnoid, which is I will show in my surgical side videos, but it is something else which is there. It is arachnida, derived from looks like spider webs. Okay, and pia is the actual covering of the brain. You know, it encompasses the brain tissue. All this, the rest of the stuff is not exactly on the surface of the brain. So, if you look at the gyral architecture, I need to show that to some yeah. So, if this is the gyrus, the pia is on the surface of the brain and it continues both on to the gyrus, sorry, it is on the brain and but it is a distinct entity. It is not that it is a mythical creature, and neurosurgically it is very important because it forms a kind of barrier for several things, and it is very important for a neurosurgical student and neurosurgeon to understand and appreciate pial boundaries whenever operating inside the brain. Okay, so that is green is pia. If you look at arachnoid, it is, as I told you, it is spider web like and can extend to varying extents to the sulci. So, there is pia and sorry, this is arachnoid and it continues like that. So, you have longer fibers, shorter fibers, things like that. So, that is arachnoid. Please do remember these terminologies, because especially for people in the biomed side, you would need to put things in context. So, when somebody discusses subarachnoid hemorrhage, you should be able to understand that it is something related to the arachnoid part of the whole system, and it is very different from an intracerebral hematoma and things like that. So, that is the idea of why I need to make the terminologies vivid, and I am taking time to explain all of these things. The last layer which needs to be discussed is the dura. So, dura is all encompassing. So, it covers over here over the entire structure. So, that is the dura. Now, dura is pretty thick in most places, excepting some areas where it is somewhat thin, and the dura forms a very tough membrane which is which encompasses the entire brain. So, now that we understand that what each one of these things mean and refer to, I think the obvious question is what happens to the rest of the space. So, there is tissue inside, there is tissue outside. What happens into the rest of the space is cerebrospinal fluid. Okay. So, I would show you in the operative videos that the consistency of the brain, it is the closest to is jelly like, the jelly like consistency. So, like you have jelly is jelly. So, it is in the sense soft. So, that is the point which I have to make, and anything soft has the tendency of getting injured, and a mechanism of protecting by understood by any mechanical engineer out there would be that you provide suspension in engineering terms, or cushioning. So, cushioning of the brain and preventing direct contact from against the skull, meninges, etc., is a function of the CSF, one of the important functions of the CSF. But the CSF is not just anything which is it is not just fluid which is there, it is a very actively secreted fluid. So, actively secreted in the sense that if you look at the Gibbs-Donan equilibrium, I had the first class, I had told about just keeping a semi-permeable membrane and putting things on either side ensures that there is an equilibrium. But like in the case of the sodium potassium pump, the CSF is generated by pumps which ensure that the constituents of the CSF are slightly different when compared to mainstream blood. So, the blood has a different concentration of various things, sodium, potassium, proteins of different kinds, whereas the CSF has a different slightly different constituent. I am not boring you with details of the CSF, neither is it would it be required, but it is important to understand that the CSF is actively secreted. It acts as a cushioning mechanism, and it is situated in this space between the dura and the pia. So, it is between the dura and the pia. Now, we need to now we have covered how many of these things? We have covered CSF, we have covered meninges, we have covered blood vessels. The next thing to be discussed about is the ventricles. So, within the brain too there are fluids filled fluid filled spaces in the brain, and those are called as ventricles. Now, the terminology is a bit confusing because even cardiac heart ventricles are named similar, and you need to actually specify which ventricle you are referring to if you are speaking to a general audience. Neuro audience would obviously understand ventricles when referred to by in a neuro setting refers to the ventricles which are within the brain. Again, anatomy would be spoken in greater detail, but the ventricles are the place where CSF is produced by structures called choroid plexus. So, choroid plexus produces CSF. So, now you would imagine that when I discussed earlier that the CSF which I spoke was between meninges outside and the pia inside, and it is within the arachnoid space. So, how the communication happens. So, that the anatomical part of the story I will tell you later, but the circulation part of the story is like this. So, CSF is produced in the ventricles, then it flows out of the brain, then it flows in the subarachnoid space, and it gets absorbed into the venous sinuses. So, there are structures called granules, arachnoid granules. See, there are controversies on many of these things. So, CSF production as such, whether it is only through the choroid plexus, whether absorption is only through the arachnoid granules, there are controversies on that. I am not including any of the controversies. The straightforward pathway which you need to understand is that this is how it happens. So, CSF, the other problem with CSF where you would need to understand in two separate engineering contexts is one, the constituents I have told you is different. It also means ionic constituents are different. Ionic, protein and nocelles. So, it is important. So, difference in all these three things happen: ions, proteins and nocelles. Now, it also implies changes in electrical conductivity. Now, it does not matter much when you are looking at any kind of electrode data which is obtained from above the dura, which is the outer membrane. But if you are looking at anything inside, you have to factor in this that there is one. Why is it important there? It is because CSF is not static, there is flow. Then the thickness sort of fluid in different parts of the subarachnoid space is different. So, both of these things have to be sort of kept in your mind when you are reading data, when you write, when you design something for recording. And you should also keep in mind that when you get data and then when you are analyzing, you may not get what you expect because of some things like this. These are not things which are routinely discussed so when you process data, but these do factor in. See, obviously if there is a change in the CSF constituent in a particular disease state, and then there is obvious electrical conductivity changes, and there is flow. So, there is flow, and so you will have to look at degradation due to degradation of electrodes, etc., due to flow. So, that is the context of discussing about CSF. Now, so the last part of the story is the skull. So, both the brain is protected by the skull, and the spinal cord is protected by the spinal column or the spine. So, both are bony structures, and I will not be discussing in detail about the spine. We will discuss a lot more about the brain, because the spine the dynamics are different, but it is not biomechanics is of course relevant, but I do not think I am covering that over here. But the brain part of the story, I am not going into the physical details of the skull, but I would need you to understand certain things in the skull for the purpose of understanding brain anatomy. It is more for understanding brain anatomy that I am teaching the skull than as an empty discussion on skull anatomy. So, the skull, which is the inner part of the within all of our heads. So, there can be deformities, there can be diseases, there can be so many things, but some things of the outer part is called the calvarium. The down part is the skull base for us. Then you have paranasal sinuses, which is in the front face, etc. This is basically for the medical side of the story, so not very relevant. And then you have got the jaw. Okay, so calvarium fairly is featureless, there is not much, but the skull base is one of the most complex anatomical structures in the body. There is a lot of stuff there. You would need to know something of the skull base, because as I told you, some anatomical entities go through various parts of it, and it is as I told you, I would also be using the skull as a basis of lot of downstream discussions. Paranasal sinuses I am not discussing, because maybe in terms of when I discuss speech, I would highlight about the role played by paranasal sinuses in modulating voice, and how sinuses help in respiration to some extent, humidifying the air, intake of air and things like that. So, that is about it, paranasal sinuses. Jaw as such we would not be dealing, it is a medical topic, I will leave it to the medical curriculum. So, most of the discussion, as I told you, will focus on the skull base, because there are certain issues, there are certain ideas which you need to understand, and these ideas are necessary when we discuss the brain in greater detail subsequently. Okay, I will break here, and then we will take it forward from the next